So thank you so much, Dr. Hutchinson, for coming in to talk to us today. Can you tell us a little bit about the type of research your lab does?
Sure. My lab is called the Multi-Scale Brain Imaging Lab because we're really interested at being able to map very small types of information related to cells and molecules across the entire brain. And we do this to investigate neurologic disorders and to develop new MRI techniques that might be promising to translate to clinical markers.
Oh, that sounds super interesting. Is this what you originally started doing in your research career, or has that changed a little bit over time?
Yeah, well, actually, my undergraduate degree was in physics and I had completely zero biology. But then I did an internship at the NIH between undergraduate and graduate school and decided that I really wanted to pursue neuroscience, but to use imaging techniques that are very sort of physics-based. And then I just loved it. And I was introduced to some of the techniques I use now in graduate school and then pursued them in a postdoc and then as a staff scientist. And now I'm here. And I think biomedical engineering has really brought together my physics background with my neuroscience interests. So that's sort of who I am and what I do.
Physics plus engineering plus biology. That's a power combo. So we know that you do really interesting MR imaging research. I'm not in the imaging field, but I know it's called diffusion MRI. Is that right?
Yeah, that's right. So my lab uses a bunch of different MRI techniques, but probably the one we use the most, just because it's the most effective, is called diffusion MRI. And actually, I have a few slides so I can take you through some of the sort of interesting aspects of that. Let's share a screen.
Yes, let's see it. Actually, and before you start, let me see if I can make sure I still have your video in here. How do I do this? Let me just experiment with a couple displays. Oh, I think, okay, we'll find out afterwards, but I think this is the right one. Okay, go for it.
Okay, so that you have my head.
Your little face in the background, yeah.
All right. Okay, so, and then I have this slide up just so that you're able to edit it easily. You'll be able to see the diffusion. Let me turn my laser pointer on too before we start.
Oh, I am so excited for this.
Really? Yeah, it's good. Actually, so I'm always excited to talk about diffusion because it's sort of this really beautiful, elegant, simple way of probing the brain. And so it's a very simple idea, which most people are familiar with when you think about a drop of dye in water, that at rest, particles are really moving around. And this has, long history. Perrin got the Nobel Prize for it in the early 1900s. And it was one of Albert Einstein's most famous papers from the early 1900s. And in fact, the Einstein equation is used to characterize the self-diffusion or the diffusion coefficient of particles. And for water, this is a self-diffusion coefficient, how much water bounces around when it's in other water. It's a very physical property, but what we've been able to do in diffusion imaging is to leverage it to be able to look inside brain tissue by sensitizing our experiments to the diffusion of water. And I want to play just a quick video to show what that looks like in brain tissue. And so what you can see is that water molecules are never truly at rest, and they bounce around according to temperature and viscosity. But in a biological tissue, the magnitude and shape of their displacement is actually related to the biological structure within which they're sort of diffusing. And so we can use this sort of physical phenomenon and leverage it to be able to measure different aspects of the shape and density of biologic tissues if we're able to measure water diffusion. And that's exactly the principle that's sort of behind diffusion imaging, where we can sensitize our experiment to measuring water diffusion in a bunch of different directions, and then use that to probe the microstructural tissue environment within, in the case of my research, within the brain. And this tells us about the types of cells that are there, what their morphologic features are. And also we can leverage it in a way to detect disorders and disease that have prominent neuropathology associated with them. And so just as an example of how we do that, most people are familiar with a whole brain MRI scan, which is more like a snapshot, and we get beautiful anatomical detail. Now, the work we do in my lab is more at this level. We're imaging small animals that we have developed different neurologic disorders in. And those brains are sort of smaller, and we get higher resolution images, but it's still just a picture. And so what diffusion MRI does, and this is sort of a long-standing technique where we've been able to use the MRI scanner in a way that is sensitive to water diffusion and map out more quantitative information that really extends into each of the voxels or pixels that you see there, so that we're really able to quantify cell morphology, for example, and other things at the micro scale using the MRI scanner. So this is a good example of a human brain stem, where you can see in the grayscale image, we can't tell much about the fiber tracts. But once we use diffusion, we can distinguish tracts like the corticospinal tract that are running up and down, or the pontine fibers, which are red here, running left and right. And it gives us a much richer description of the neuroanatomy inside the brain. We can also use advanced techniques to measure tracts through the brain. This is tractography, which looks nice, and most people have seen looking at white matter tracts using this technique. And something that my lab's really interested in is leveraging these techniques in different ways that give us quantitative markers, almost like MRI stains of tissue that might report different biophysical information about either normal tissue or pathology in the brain. And this is an example of Laurel Dickhouse's work. She's a PhD student in my lab in biomedical engineering, but with a minor in neuroscience. And we've partnered up with Carol Barnes' lab to look at some of their bonnet macaque brains using these techniques at very high resolution in an effort to understand what the different types of MRI scans can offer us in terms of representing different tissue environments. And Courtney Comrie in my lab, my other PhD student, also in BME, but with a background or with a minor in electrical engineering, has been using these types of techniques to look at human tissue with the goal of translating them as clinical markers. And these are in Alzheimer's disease postmortem specimens. And she's been able to compare some of the more advanced diffusion metrics to conventional ones to see if we can sort of probe more information and develop sort of more sensitive markers to AD pathology. So that's sort of what I do in a nutshell. And I hope I've represented diffusion MRI. It's a very old and beautiful technique, but it also is at the cutting edge because people are still developing new ways to use it, new mathematical models to represent the microstructural environment. And I think it's a really promising direction for developing clinical markers. I do have a talk on sort of more details about this. I recently gave a talk to the Martinos Center at their Brain Map seminar, and they posted it on their YouTube channel, and that link is right here. So I hope that's a good overview of diffusion MRI.